webinar. It's 1 o'clock noon here in Berlin. We are physically hosting the event, but I would like to say hello and good morning to all of our participants joining us from Asia, Australia, Europe, and Latin America. Thanks for joining. My name is Robertus Pietsch. I'm the head of MR and CT Contrast Media Research Department here in Berlin. And together with my colleague, Dr. Laura Schöckel, Medical Clinical Affairs, we are delighted to guide and moderate the session today. Our goal is to consistently provide you with the latest scientific information, and especially today with exciting scientific studies on the newly discovered G-lymphatic system. We are therefore very pleased to welcome Geir Ringstad. G-lymphatic MR imaging, the new kid on the block, is the topic of our webinar today. Geir Ringstad is a neuroradiologist and researcher in the Department of Radiology and Nuclear Medicine at Oslo University Hospital in Norway. Dr. Ringstad's main research focuses on novel methods for MR imaging of G-lymphatic and lymphatic brain clearance. On the occasion of the 40th Congress of the European Society of Neuroradiology in Oslo, he presented the scientific results together with other outstanding experts. This lecture was very well attended and was highly appreciated by the radiological community. I was attending and I was very excited and convinced that this new field will be of high interest in the future, and also the response of the audience here of our webinar will hopefully the same. Through extensive research and the publication of numerous studies, Geir Ringstad played a key role in the elucidating the mechanism and future possibilities of G-lymphatic MR imaging. This MR technique could be a new tool for the early diagnosis of neurodegenerative diseases. The key learnings of our webinar will be to understand the importance of the G-lymphatic system and the clinical relevance of neurological diseases. And in addition, G-lymphatic contrast enhanced MR imaging as a new clinical application in the central nervous system. Before we start, I would like to be aware that Dr. Ringstad's presentation will be followed by a joint Q&A session. So please send your questions via the webcast chat function that you can see on the left of your screen so that we can take them up in the discussion session. So with this, Dr. Ringstad, the stage is yours.
Thank you, Professor Pietsch, for that very nice presentation. And hello, everybody. I'd like to say good morning and good afternoon, and perhaps it's even good night somewhere. I'd, of course, prefer to see all of your faces, but on the good side, I think this format of presentation might reach even more of you. So that's very good. So we will start out now with talking about imaging of human brain extravascular pathways and molecular clearance function assessed with CSF tracers. And that's long, and in short, we might call it lymphatic MRI. And I guess that many of you attending this webinar today are somewhat familiar with these concepts. And I'll try to dig into what has been done previously in basic research, and then go on to tell you some more about our experiences with human studies. This is one disclaimer, and this is my conflicts of interest. And let's just start off with this paradoxical situation, you might say, because I guess we're well aware that the brain only accounts for about 2% of the body mass, but still demands up to a quarter of the body's energy supply. And waste metabolites in the brain are constantly produced as we speak. And if you see this map to the left, that's a typical atlas image of the anatomy of the lymphatic system. And if you see above your collar, there is no lymphatic vessels within the intracranial compartment. And as you know, the lymphatic vessels are very important for clearing toxic waste substances from the entire body, except how is it within intracranial compartment? And that's quite an enigma. I mean, there are some transporters over the blood-brain barrier. For instance, amyloid beta 40 isomer is cleared over the blood-brain barrier. So there are some excretion routes that are already defined. But for some molecules, including some amyloid beta isoforms and tau protein, which are neurotoxic to the brain and might cause Alzheimer's disease, there have not been any identified transporters over the blood-brain barrier. So how are these substances cleared? And an answer to this paradox was given in 2012 when the Nedergaard group, is a Danish researcher, but from Rochester, New York, when they published this seminal paper, I must say, in Science Translational Medicine in 2012, because here they provide a new concept for how waste solids from the brain are cleared along vessels. As you know, and I cannot point on my screen so you can see it, but you know that the brain is surrounded by cerebrospinal fluid everywhere. And in this concept of the G lymphatic system or lymphatic system, CSF enters the brain and communicates with the paravascular spaces along arteries. And when the CSF enters along arteries, it traverses down into the cortex. And here you will find the astrocytic end feet that surround all vessels, and particularly the arteries. And at these astrocytic end feet, you find a lot of water channels or aquaporin 4. And in the lymphatic concept, water permeates these channels, go into the interstitial space, and flushes with it these toxic waste products that are constantly being produced by the cells of the brain. And then the water moves these waste metabolites further out along the veins, which you see to the right in the lower image, and then out of the brain along the veins. And what is important here, and what is still controversial, is that this is more than just diffusion. They talk about convection. So this is net flow of CSF driving the metabolites from the one end to the other. And the driving force for this propagation of fluid and molecules is the pulsating arteries at the surface of the brain. Because you know the arteries, they are pulsating, and the pulsations are mediated through the cerebrospinal fluid, which again are mediated into the perivascular spaces. And it's been a lot of scientific discussion about that, whether the pulsations might be mediated deep into the perivascular spaces around arteries, and whether there is convection at all or only diffusion. And that controversy hasn't quite been solved within neuroscience yet. So this is another image where you see water channels, and here they look quite abundant. So there are very many water channels, and to the left here you see an electron micrograph of water channels, and they cover up to 40% or even 50% of the astrocytic end feet area surrounding all vessels. So that these aquaporins should have an important role for flow of water is obvious. And when the Nedergaard group did their experiments, they did their experiments on mice. And in the image to the left, you see a mouse with its aquaporin 4 intact. And to the right, you see the aquaporin 4 knockout mouse without the aquaporin. And here they injected a fluorescent tracer in the cisterna magna, and this is the effect you see from knocking out the aquaporin 4. So it's much more penetration of tracer into the brain from surface in the wild type mouse compared to the knockout mouse to the right. So that was interesting. So already indicates that aquaporin 4 really matters for how a tracer from CSF compartment at the surface can enter the brain. And then it got even more interesting because they looked at the same cohorts of mice, the wild type and the aquaporin 4, and then they injected an amyloid beta tracer into the parenchyma. And then you see on the graphic illustration to the right that aquaporin 4 knockout mice have a quite slower excretion of the amyloid tracer than the wild type mice. So aquaporin 4 status also is important for the excretion of amyloid beta from brain tissue. And it got even more interesting, and this reached widespread attention when the same group published this paper in Science 2013, when they could show that the lymphatic system is actually active only during nighttime, or about only during nighttime. And this is due to a contraction of many of the glia cells, allowing for an increased volume of the interstitial space where these molecular motions occur. So when these mouse were sleeping, this allowed for a doubled rate of excretion of amyloid beta, and they found a 60% or more than 60% increase in interstitial space. So this provided an explanation that many have been looking for why you need to sleep, because sleep clears your mind. But this study has never been reproduced, I think, in a higher species, and not in humans yet. But still very, very interesting. And then they associated aquaporin 4 status with cognition and Alzheimer's disease in aging brains, and found that the loss of perivascular aquaporin 4 localization was one of the factors that rendered the aging brain vulnerable to amyloid beta aggravation and neurodegeneration. And they also found that this perivascular clearance, or the lymphatic clearance, declined with aging, still in mice and lungs, and also with disease, other disease, like subarachnoid hemorrhages or ischemic stroke, which significantly impaired lymphatic system perfusion. So this has quite widespread implications for a lot of brain diseases. And not to forget traumatic brain injury, where post-traumatic mice had far less entry through perivascular spaces or tracer after trauma. So the same group, and the first author is Jeff Eilish, and this is the Neville group. They did their first experiments with intrathecal contrast agents on rats. And what you see here is the color-coded degree of enhancement in rat brain, and you see the time curves at the lower end of this page. And they see that first, the contrast enhances around arteries and then goes more diffusely into the brain parenchyma. And from this, they also suggested that the lymphatic pathway function also could be assessed in humans by using MRI contrast agents as a CSF tracer. And then we come to our research, and this is what we have been doing in a study setting since 2015. And since 2015, we have enrolled 149 patients with this methodology. We have done this on patients who are under workup for suspected CSF disorders. We got approval from the National Medicine Agency, the Regional Ethical Committee, and the hospital authority for doing this. So we have not done this on any healthy subjects. We published our results of the safety study of the first 100 patients in the AJNR last year. And you see here that we had no serious adverse events. There were non-serious adverse events, mainly headache and nausea. For these patients, we also administered iodixonol or Visipac in conjunction with gabapentin, just to assure for correct needle position through the lumbar injection. And the side effect profile was quite comparable to Visipac or iodixonol alone. So this is more schematic outline of our technique for what we call GMRI. You do first pre-scan MRI, including T1 volume sequence, and then the patient is given 0.5 milliliter of gabapentin, and then we started imaging immediately after, and then this went on to the next morning, 24 hours after, and then to the next morning, 48 hours after, and we also have seen somebody after 72 hours, and then everybody came to a 4 weeks follow-up exam. So this was the methodology, and this is the type of images that you get out of the scanner. So these are T1 volume scans, pre-contrast to the left, and then you follow the timeline towards the right, and we have included the 24-hour time points. So this is interesting, and you already see some of the distribution of the contrast agent as CSF tracer within the ventricular compartment and the surface and so on. But I would like to then address this question, because what should we really expect? I mean, should we expect brain tissue enhancement as they did in the rat studies? And that was not readily to be expected, because there are reports from the basic sciences, and this was in Nature Immunology 2017, where it is stated quite clearly that in both the human and rodent cerebral cortex, there is no perivascular space. So there is no, in theory, you should not expect that your contrast agent would have access to perivascular spaces in the cortex. But mind you that this is based on fixed specimens, human and mouse brain specimens, and then they reveal no such space. We know from other reports that perivascular spaces have a tendency to collapse under fixation. So this could also, in somebody's opinion, be due to an artifact, but we did not really know what to expect when we really looked into these images. And this is from another tracer study where they injected into CSF an amyloid beta tracer, and the tracer penetrated just immediately below the surface, like 0.2 millimeters, as you see here, which is almost nothing. So in the hands of these research groups, you would not expect to find a CSF tracer within brain tissue. So this is what we find. And this is one human subject without hydrocephalus and the workup for another CSF disorder. I think it was perhaps CSF leakage. And here you see on the color-coded scale, that is the percentage T1 signal increase within brain tissue. So here we subtracted all signal from the CSF at the surface and the ventricles. So this is enhancement from surface in brain tissue. And you see, after a few hours, you see in the column 69 hours, then the enhancement is starting to appear quite obviously. And there are some things to be noted here. First, you can note that the tracer enhances mostly around your large artery trunks within at the brain surface. So you have enhancement nearby or adjacent to your middle cerebral arteries in the insula and around the operculum. And you have enhancement in the medial temporal lobes, which is in the hippocampus, entorhinal cortex, hypothalamus, which is close to the circle of Willis, and the posterior cerebral arteries. And you also have enhancement in the cingulum, which is adjacent to the anterior cerebral arteries. And then the enhancement after 24 hours is much more diffuse. So then it's quite brain-wide, and we found actually enhancement in all free surfer subregions of the brain. So the contrast, it had access to all parts of the brain. So we found that very interesting, and this is also what you could expect from if you give another drug intrathecally. Might be for diagnostic or treatment purposes. If this drug would share the properties of our CSF tracer, which was scan control, then this is also what you might expect of the fate of your injected drug. And of course, we have for many years used off-label MRI contrast agents for workup of CSF leakages, but we did not think of acquiring these very late images after 24 and 48 hours. So when we didn't look for this, we had no opportunity to find it either. So this is interesting, and of course, we would also like to connect our findings to the dementia population. Is there something here that reflects the theories from the Rodden studies with the amyloid beta, decreased excretion rate, and so on? And we would like to just note about the association between this enhancement pattern in brain and also how an amyloid pathology propagates through brain. These are not exactly the same, but many of the same areas that you see amyloid and tau pathology in the early and late stages, actually. So from this, we're going to just one comparison with the animal studies. So you see that, like in a wild type mouse, in the upper of this slide, upper part of the slide, you see that you have quite brain-wide enhancement of the tracer, and so do we find in our human studies. To the right, you see also that we did our MRI follow-up up to 4 weeks, and there was no sign of any tracer at all in the parenchyma. We did this also to look for any signs of retention, and then I would actually like to go backwards one slide here, because there's also been a lot of discussion about the routes for gadolinium into the perivascular compartments, which has been demonstrated after intravenous injections, and particularly with linear agents. And of course, we know now from previous research that after intravenous administration, the CSF or the MRI contrast agent leaks into CSF at the different regions, and perhaps particularly through the choroid plexus. And then this might be transported with the CSF and circulate around the brain surface and then enter the paravascular or lymphatic pathways. And that might actually be a route for entry of gadolinium into the perivascular spaces of the brain after intravenous injection. Okay, so now look at the upper panel again, and we would like to think, what do we actually see in these images? Because we try to dig a little deeper into it. And I think we must acknowledge that there is also a lot of diffusion going on here. There is no reason to believe that diffusion does not go on all the time, as in the entire brain. But at least we know that the gadolinium molecule is hydrophilic, and it's quite small. It's like we estimated less than 2 nanometers, which is very small. And then at least we know it may be entering the paravascular spaces, and that is also, I think, probable, because when you see the enhancement around the pulsating arteries at the surface, that speaks in favor that pulsations really matter for the tracer to enter the brain. So we think that much of what we see in the early scans on day 1 is mainly tracer going into the periarterial spaces. And then this small tracer is not expected to enter the interstitium through the apopores, because they only allow water and a few ions. But between the astrocytic end-sheaths, you have astrocytic end-sheath gaps, and they have been reported to be up to 20 nanometers. So it is quite probable that also the gallibutrol as CSF tracer may enter from the periarterial perivascular space into the interstitial space. And this is also something I was quite fascinated to observe, and this is after 6 hours in one patient, but we also saw it in several patients, that during the first hours of distribution of this CSF tracer at the brain surface, it seems to follow the leading paths of the great artery trunks at the surface, and before it gets more widely diffused and widespread. And you also see this very circular pattern, which seems to delineate the artery wall. And I've just tried to speculate what this really is, but if you look at the cartoon to the lower right and the electron micrograph at the upper right, you see that the leptomeningeal cell sheets that encircle the arteries at the brain surface, they have these openings, or what they call stomata. And it might be so that our tracer also enters the perivascular space around the arteries at the brain surface before they are funneled into and penetrates the brain from surface. So it's just an observation that we just try to explain what we see. And then I would like to tell you something about normal pressure hydrocephalus, because this is a dementia group, and these are quite a proportion of the study cohort that we examined. And as you probably know, that normal pressure hydrocephalus has a great overlap with Alzheimer's disease regarding amyloid and tau pathology within brain tissue. So this could also be a similar model to Alzheimer, at least with regards to amyloid beta and tau. And the NPH population typically also have restricted pulsatility intracranially, and in our hospital they are also being investigated with overnight intracranial pressure monitoring, where we find these restricted pulsatility patterns at ICP. So we investigated these patients and we investigated them with MRI. And this is what we observed. So here you see the baseline scan to the left with the ventriculomegaly. And then we go on to look at the 69 hours and the 24 hours. And this is similar. This is also brain-wide enhancement. But also note that we now have the enhancement around the ventricles. And this is due to the ventricular reflux. And a very common pattern in these patients is what you see to the upper right corner. It's early and quite large ventricular reflux of the complex agent. And to the lower right corner, you see actually directly the complex enhancement enhancing in the paraventricular white matter, which corresponds well with flare hyperintensities. So this is one observation from this NPH group. But then we try to compare them with the references. And the references, they were patients under workup for other disorders, but without dementia. And here you see that the excretion of our CSF tracer is delayed from both CSF and the cortex and the white matter. You might read the entire paper. I gave the reference to the lower right, but we found this from a range of brain regions. So here we can see, I think this was for perhaps the first time, that a CSF tracer administered intrathecally is delayed in a dementia group compared to a reference group. So this was interesting, and this echoes with some of the findings from the animal studies. But there are differences. So if you now look at the timelines, at the bottom you see the time curves of Jeff Eiliff and co-workers from 2013. And at the upper row, you see our studies. And look at the very, very different timing of events. In rodents, this is more or less done in 2 or 3 hours. But in humans, this is a much more durable process, and you need to follow it for a much longer time to catch these events. And you see that particularly perhaps after 24 hours, you see the biggest difference between the NPH patients and the references. So what decides the clearance from CSF? Well, in 2015, 2 independent groups first or rediscovered true lymphatic vessels in the dura, in the meninges. It was the Kidneys group in Virginia, USA. It was Asplund and co-workers from Finland. And they described dural lymphatic vessels, true lymphatic vessels, that communicate with the cerebrospinal fluid. And it's also been a later publication that you see to the right, which is quite fascinating, and I recommend you all to look into this. It is still as a preprint, so it's not fully peer-reviewed yet, but this is from a group in Chicago, Illinois, and then they have found lymphatic vessels both in animal and human meninges. And a very important observation here in all of these studies is that if you occlude these lymphatic vessels, and if you do it at the level of your neck, then amyloid accumulates in the meninges and also in the brain. So brain clearance of toxic waste products like amyloid and tau seem dependent on normal lymphatic meningeal clearance. And this is very important. And they also have shown that with aging, your lymphatic clearance capacity declines. So we also would like to investigate this further with our CSF tracer. This has implications for a lot of things. So this is T1 black blood. This is T1 weighted, but it's black blood. So signals from flowing vessels. And this is with gallibutrol intrathecally, which is enhancing the CSF spaces. And you see very well to the left that we find these molecular efflux routes going out through the basal neuroforamen of the skull. So, and that's been described recently, that you have molecular efflux through lymphatic pathways along your cranial nerves. But we also found something else, and that's just out of our images first, and then you see the arrows indicating something enhancing along the superior sagittal sinus. And this is in the vicinity of where they have described functional lymphatic vessels in animals. So we were particularly interested in this region. And it's the same what you see from the flare. I guess many of you who are radiologists have acknowledged that you have some hyperintense regions at the parasagittal dura in this area along the superior sagittal sinus. It's most prevalent along the middle third of the superior sagittal sinus. And we went to the anatomy books and literature to see, have anybody described this yet? Because it has been some critique about lymphatic vessels, because it's supposed that the arachnoid membrane is impermeable to molecules and even water. So in principle, a CSF tracer should not go through the arachnoid. So should we expect then to find a tracer in the parasagittal dural tissue? Our image resolution is 1 millimeter, so we had no aspirations to find these small lymphatic vessels themselves, but could we find an enhancement in the vicinity of these vessels? So this is a cartoon from an 96 anatomical description where they show here in red the small intradural arachnoid granulations. And then you also have all these venous lacuna, and they speculate in this paper that these might be the pathway from CSF through these and back to the venous circulation. Well worth reading this paper as well, so I don't have the time here to go into the details. And this is our images. So we start up on the upper left corner with T2 flare. Then you see this tissue that is not veins because it has high signal, and it's not CSF because of this high signal flare. And then you have the T1 black blood pre-scan, upper middle row, and then you also see that this is not veins because it is not known by this technique. So then we observe this particular region after some time, and then you see enhancement here. So this is enhancing parasagittal dura. You see it's quite associated with the CSF signal increase, but not the same. Particularly at 48 hours, you see it quite differently. So we think this is exciting. We got to publish it this year in Nature Communications, and we think by showing this, there is a potential for also this communication in humans between the CSF and meningeal lymphatic vessels, because meningeal lymphatic vessels, they do not have any tight junctions. So if you already have your CSF tracer in a parasagittal dura, the distance or your pathway towards the lymphatic system is very short. And this is very nice images made by my colleague, Thomas Akinis. And you see that at late scans, in the middle here you see 48 hours and also to the right. the tracer molecule seems to almost migrate in regions adjacent to the paratityl dura, which is here colored yellow. So this might be a clearance route. To which extent this is dominant or very important, we do not know because we haven't really quantified it. But we have identified that the CSF tracer actually enhances and accumulates in the paratityl dura on these late scans. And this is perhaps also important for acknowledging the communication with the immune system at the lymphatic vessels and the lymphatic system. And this might go both ways, because if the lymphatic vessels are important for clearance of amyloid beta from the brain through the CSF, it might also be backwards, that lymphatic cells and substances like cytokines and interleukins and so on may be excreted by the lymphatic vessels into the CSF as a carrier medium, and then is distributed brain-wide through the CSF. So it's been an enigma, as many of you know, that how the brain and immunological diseases occur and how it occurs together, because you do not have true lymphatic vessels within the brain. So the communication between meningeal lymphatic vessels and the brain through the lymphatic system might be worth looking more at in the future. And the Kipnis, the war group from the US in particular, have done very important groundwork on this, so I recommend reading some of their papers. So what might be the future project when we now are closing up here? Well, you know that in Alzheimer's disease, it's typical that neurodegeneration already has occurred when you start to get symptoms. Then already the amyloid beta and the tau pathology will be present in the brain. So ideally, you would like to look for pathology somehow before symptoms occur, before neurodegeneration occurs, which is irreversible. So we think that might it be so that measuring clearance of a CSF tracer might identify neurodegenerative disease before the symptomatic phase. And we look particularly here at this region, and the arrows, the lower, the stippled arrows, is toward the entorhinal cortex. The entorhinal cortex is the first site of neurodegeneration in your brain. It precedes hippocampal atrophy. This is the same area where you have your grid cells, which are cells that code for your, it's like your brain's global positioning system. And something in the first that you lose in Alzheimer's is your sense of direction, and you get lost. So looking at molecular enhancement and clearance capacity from this region might be interest to detect early reduced clearance capacity. So we have suggested this in this paper. And this is also quite interesting, because we thought what you really want to know is the clearance of molecules from your intracranial compartment to blood, because that's where also the amyloid beta and the tau ends up finally. So here we have retrieved a lot of blood tests from these patients, and we compared the blood profile of galabutral after all these time points. And if this worked, then perhaps you wouldn't need imaging. So then you see here to the left, we have CSF leak patients in red, and you have the reference cohort in blue. And here you see, this is also almost a demonstration of CSF leakage, because you see in blood, you see an increase much faster than in the references, which were patients ending up having no treatable pathology. So to the left, you see CSF leaking faster into blood than in the references. But if you go to the right, then you see the opposite. The references are still in blue, but now the red is the dementia cohort. And what we see here is that you might inject an intrathecal tracer molecule and then measure delayed clearance from your craniospinal compartment to blood. And there are several time points here where you see statistically significant differences between these two groups. And these are only from a few patients, and still we found the effect. And of course, ideally, you would see this in single patients. And then you will perhaps have one test to identify decreased craniospinal molecular clearance rate preceding cognitive decline. And another potential future prospect we thought of is that, a lot of these brain pathologies that we image every day in radiology, we do not have so much utility of intravenous agents. It's just when you have a blood-brain barrier leakage, then you see enhancement, of course. But then otherwise you don't get so much out of using your contrast agent, of course, for perfusion as well. But so we talked about how is it if you inject a tracer and then enhance the entire brain outside the blood-brain barrier and not within. And this is work in progress, and we don't know that. But for instance, both with glioma disease and multiple sclerosis, there are more pathology than even high-field MRI can image. And could this methodology allow for detecting more pathology? That we don't know that yet. So I arrive at some conclusions here. So let's say that the glymphatic system adds also a metabolic and immunological dimension to cerebrospinal fluid flow. It also provides a link between vascular health and brain health, because healthy pulsations is important for healthy propagation of CSF pulsations through the brain. And this is also clinically relevant to a range of common neurological diseases and sleep. And we know that we can visualize paravascular molecular pathways with intrathecal contrast-enhanced MRI. And we know that this lymphatic capacity for clearance of toxic metabolites may be assessed by administration of extravascular CSF tracers. So with this, these are some of my co-workers that I would like to acknowledge. And with this slide, I think I would like to thank you for your kind attention. I think I used perhaps 5 more minutes than I promised, but one gets inspired. So that's what it is. So I'm ready now to take your questions online. So then I'll give the word back to the moderator. Thank you so much.
Thank you very much, Geir Ringstedt, for your very, very exciting presentation today. So, and I'm sure we are all aware now that the brain has its own waste clearance system, the so-called glymphatic system, and that sleep is much more important than expected to clear out all the waste products, such as amyloid beta, tau protein, or probably even toxins, such as alcohol from the brain. Importantly, and this is what you showed us today, is that contrast-enhanced MR imaging of the glymphatic system and CSF can be done to assess the glymphatic function, and that delayed CSF flow could be an indicator for dementia. Therefore, glymphatic MR imaging could be a new clinical application and a new tool for the early diagnosis of neurodegenerative diseases before symptoms may occur. So thanks again, Geir, for all the insights, and let's start with the Q&A session. Therefore, please send in your questions via the webcast chat function that you can see on the left of your screen. And I've seen that some questions are already sent in. Let's start with the first one. Apart from giving contrast, could any other sequence visualize the system?
Perhaps. we are about to start working on this. Of course, if you could find a non-invasive marker of the lymphatic system, that would be preferable. Now we consider MRI as like gold standard because then you actually follow the molecules. But there has been some approaches, by instance, using DTI. So if you go to PubMed and you write DTI on the lymphatic system, then you'll get some hits there. So that's one possibility. And of course, this also has some very interesting implications with what are the meaning of enlarged perivascular spaces that we see in our images all the time. So do they really communicate with the subarachnoid compartment or do they not? So what's the implications of finding enlarged perivascular spaces? So it's a growing body of literature on these matters. So I would encourage all with interest in these things to couple your interest field with lymphatic system on PubMed, and you will get a lot of exciting stuff.
Thank you very much. Another question. Can you please tell the dose of gadobutrol you give?
Yes, for most patients we have used 0.5 milliliter. We have also, for some patients, tried to lower the dose because that would be preferable. So we also have used some patients with 0.25 milliliters. And that might also work as well. Then, of course, you have a little less enhancement. And we are about to publish our safety study on the full 149 patients cohort. And then we will compare the safety profile of 0.5 and 0.25. Yeah.
Thank you. So we still have a few minutes for questions, so please type them in. Another question, do you believe that the sensitivity of today will be sufficient to describe changes in amyloid beta deposition?
I guess then they mean the sensitivity of this current method, if it is sufficient to demonstrate amyloid beta decreased clearance. Is that right?
I think so, yeah.
Yeah. Well, that's a good question because, when we administer gadobutrol, that is a surrogate tracer for amyloid beta. The molecular weight of gadobutrol is 604 Dalton. And if you look at amyloid beta, it's much larger, like 10 times larger. And if you look at the tau protein, it's even larger. So what the benefits of using gadobutrol is that it accesses the entire brain. It is hydrophilic, and it does not react with other cells and molecules. So it's, I think the utility of it as a CSF tracer, then it's better to have a small tracer than a too large tracer, because then you wouldn't see so much. But still, we have to keep in mind that this is a surrogate marker for the paravascular excretion of these other molecules that we are really interested in, which is amyloid and tau. But of course, we do not have any such tracers, and I don't think we would give them either. So that's a weakness with our approach, that it's not quite similar to these other molecules.
Thank you. So the other question is, does glymphatic MR imaging work with intravenous injection?
Interesting. I'm happy for that question because I mentioned DTA as a non-invasive marker, but you should also look at the studies from Alexander Radbrook and his group. He's presently in Bonn, and they have presented some very, very interesting studies, for instance, in investigative radiology, where they have administered intravenous contrast, and then they have used dedicated imaging to detect these small amounts of contrast leaking into the CSF and try to make the most out of it from that end. I'm not sure if that is sufficient amount, but these approaches are anyway very exciting, and I think they are worth following up. So I'm in contact with Alexander Rabrox now, and we are thinking about some collaboration efforts. So I think we will see more of that in the future.
Sounds exciting. So another question, what are the technical requirements? So do you perform your study only at 3T, or what are other technical parameters that you're using?
Yes, so far all our patients have been done on 3 Tesla on a Philips scanner with T1 weighted imaging. We have not done any on 1.5 Tesla. I guess that would not be such a big issue, because what you really want to see is any relative changes. And I'm quite convinced that at least assessing the tracer clearance within the CSF is important, because we always see that the enhancement of the brain is very highly associated with the enhancement of the CSF. So I think the CSF clearance really matters here. And that also speaks for the importance of the lymphatic and meningeal vessels and these clearance routes. So even though I haven't tried 1.5 Tesla, I think for many of these matters, for estimating clearance, you should come very far also with the 1.5 Tesla. But this, of course, is a research setting, and we haven't started doing these strictly on clinical grounds yet.
Another question from the audience, what sort of preclinical animal tracers might work well to see the lymphatic flow? Are there fluorescent tracers which might work well?
Well, preclinical, then you mean animal studies. Then I would just refer to the studies of the Nedergaard group with Jeff Iliff and co-workers. Then they have used fluorescent tracers for their fluorescent imaging, but they also used MRI contrast agent through intrathecal injections in rats. And then they first used manganese, which is linear, and we have stuck to our macrocyclic agents, gadabetrol, and now I see also the preclinical researchers have converted to gadabetrol, which I think is a good idea.
Thank you. So one very last question. Are parasagittal meningeal lymphatics proved by histological studies?
Yes, they are quite convincingly proved. The publication from 2015 from Antoine Louveau and co-workers, that was in Nature Medicine, and they have imaged this with a specific coloring where they detect this, they call it pre-lymphatic vessels, but there are lymph vessels there. And if you go to the paper that I showed you, the Romanova and co-workers from Chicago, Illinois, then they find that actually there are meningeal lymphatic vessels in very many parts of the dura. And this is some sort of a rediscovery, because it seems to have been meningeal lymphatic vessels there, always. And it has been described much previously. But the importance that really grew was that you saw that a tracer from CSF passed into these meningeal lymphatic vessels. Previously, I think they must have been thinking that they only are lymph vessels for the meninges themselves. But if these also are meningeal lymphatic vessels draining molecules from the CSF and the brain, then these vessels would really matter. So I think that discovery really lifted the attention for this announcement.
Thank you very much, Geir. Unfortunately, we are really coming to an end. The 1 hour is over today, and I would like to close by, of course, thanking you, Geir Ringstadt, once more for your effort, your excellent talk, and also many thanks to all of your active participants. I think we have had a very productive and exciting discussion today, and we will all answer the questions that came in individually that we did not answer right now. I hope you have all enjoyed the science as much as I did, and we would appreciate if you could complete the short survey on this webcast that appears at the end, and your feedback will inform future programs. So I wish all of you in Europe, Australia, Asia, Latin America a very nice day, morning, evening, and to our participants here in Berlin, a good afternoon, and thank you all, and stay safe and healthy. Goodbye, everybody.